DNA replication is the process of producing two daughter strands that are identical to the one original DNA molecule. We will show you what happens with both of those strands. First, the enzyme helicase comes in and works to unwind the DNA double helix into two separate strands. The DNA helicase creates a replication fork. The fork makes a leading strand and a lagging strand. Once they are unwound, another enzyme known as RNA primase enters starting at the 5' end of the leading strand. This enzyme creates what are known as RNA primers on the original strand of DNA that tell DNA polymerase III where to start replication. Once the primer is set, DNA polymerase III brings in the new strand of nucleotides that are complementary to the original strand. These nucleotides are attached to the original strand using smaller proteins. Then an enzyme known as DNA polymerase I comes in and removes the RNA primer so that it can be replaced with DNA. Thus, the leading strand is completed. The lagging strand begins the same way as the leading strand, only with this strand, the RNA primase, starting at the 3' end of the original strand, places primers at multiple places along the original strand. Once the primer is in place, the DNA polymerase III returns starting at 3' and adds the new complementary strand of nucleotides. Then the DNA polymerase I comes in behind the DNA polymerase III and replaces the primers with DNA. Next, an enzyme called ligase will come in and seal gaps in the phosphate backbone. Thus, the lagging strand is complete. Now you have two daughter strands of DNA that are completely identical to the original.